know him very well, but still, as customary, I will introduce of All India Institute of Medical Sciences, New Delhi, and currently holds the position of ICMR Emeritus Scientist. Earlier, he served as national chair for 5 years, and he was professor and head of Department of Transplant Immunology and Immunogenetics at AIIMS. In fact, he is the founder of that department a long time back, and he carried on as for a long time. So he built the department. It is one of the finest in the country. Professor Mehra is a fellow of many national and international academies, National Academy of National Science Academy, Indian Society of Organ Transplantation, member honorary COSA of the Hungarian Academy of Science, and fellow of the World Academy of Science. He's recipient of several awards. I will not really read all that because it will take 10-15 minutes to read all the awards, but very significant one is the Shanti Swarup Bhatnagar Award he got, and then Ranbaxy Foundation Award he got, and many other prestigious awards. His research domain, everybody knows, ranges from investigating the immunology of infectious and autoimmune diseases, potential role of HLA matching in organ and hematopoietic stem cell transplantation, influence of alloantibodies on transplant surgery, and all. So he mastered this transplant immunology and brought it to the level that it is now considered one of the best ones in the world. So I invite Professor Mehra to please deliver his talk on COVID-19 and host immunity. Professor Mehra, please.
Thank you very much, Sunil. I must first of all congratulate you and Amit. Both of you have really got together to do this course, and I'm sure this is going to be very helpful for our young scientists. And I was here partly yesterday, and I found that the course was really very absorbing. It is really a very nice thing to do. And in COVID-19 era, I think it has become a norm now that we do such kind of conferences or the courses online. Now, I'm going to speak to you about COVID-19 and host immunity. A lot of people ask the question, when will the pandemic end? I really don't know when will it end. Only the astrologer can probably tell. But we'll talk about some of the developments that have taken place in the last 8 or 9 months in the field of immunology or vaccines. Now, you see the pandemics over the centuries. If you look at the pandemics over the centuries, beginning from 6th century or 14th century, when plague was the most common one, and you have learned about the Black Death and number of people actually dying out of it. Smallpox was the main thing in the 16th century. And then you come to the 20th century. In the last century, you saw a very high pandemic, the 1918 flu pandemic, also called as the Spanish flu at the time. Nearly 50 to 100 million deaths have taken place over there. In the 21st century, in just the 20 years that we have seen the 21st century, we have already seen 3 episodes of coronavirus infection. The number one was in 2002, and that the name was given as SARS. It was now not called SARS-1, it was just given SARS, SARS epidemic. And the virus, of course, started from Guangdong, China. And the total of 8,422 known cases were known, and with 916 deaths. And that makes the fatality rate of 10 to 11, which is really very high. The peak was in May 2003, and WHO declared the threat over by July 2003. So it was really there for a very short time. Another was in 2012, MERS, the Middle East outbreak, and the total cases that we know, recorded ones are 2,494 with 862 deaths. And you can see the fatality rate in this one was even higher, 34.5. And it was the first reported in Saudi Arabia. In fact, it was mostly confined to the Arabian Peninsula and mostly to at least Saudi Arabia. Then, the one that we're talking now, we have seen that over 1 million deaths have already taken place. This is the 2019-20 pandemic. The fatality rate of this one is rather low. So why actually blame him so much? But you will see that the infectivity rate of the virus is very high. So word over, the fatality rate is very low. In India, of course, it is only about 1.45, but it ranges from 1.4 to 1.7 or so. But then it is all the time going down as we are testing more. This is the SEM picture of a cell from a patient sample heavily infected with SARS-CoV-2 virus. And you can see the yellow ones on this cell. And the ancestral type O is known. There are 11 subtypes are known. The most prevalent subtype is the A2A. And the A2A subtype is found in almost all countries around the world with D614G substitution. This substitution of aspartic acid to glycine gives it the ability to become even more infective because it can now attach to the AC2 receptors on the epithelial cells. It gives it a robust anchoring feature and ability to enter the host cell. What we have seen in recent years, or recent months rather, I would say, or recent weeks, role of linoleic acid in RBD of spike protein, the receptor binding domain. This is a very important study, and I'll talk to you a little bit about that towards the end of my talk. Cryo-EM shows that linoleic acid is most likely the molecule to which the virus attaches to enter the human body. So we are actually learning about the virus almost every day. Look at the relationship between viruses, animals, and humans. All of these three viruses, as you can see, have originated from bats. At least we know until today that they all originated from bats. But the intermediaries, as you can see, varied from 2002 in civets to camels, and now people talk about pangolins. It must have gone through the pangolins to the humans. So it's like a jump species from bats onto the humans, and we know that it is through the droplet infection. And that raises the question for us immunologists, how do bats survive coronaviruses, as both can coexist for long term? Do bats have a unique or a super immune system? But we have known that under stress, the balance between the bats and the virus can get disrupted, causing the pathogen to multiply, mutate, and transmit to humans. This seems to be the most plausible explanation that if we are talking of Wuhan as the center of the virus over there, maybe there was some stress on the bats, so then the virus got disrupted, mutated, and actually got transmitted to the humans. But there are many theories, many more theories. I really don't want to go into details of any of those. I am very fond of looking at this John Hopkins dashboard every day because this is a beautiful dashboard. It gives you infection numbers. You can see over here that the numbers today, we are talking about 36 million around the world, and gives around over here the countries, about 22 of them with more than 300,000 infections. Of course, India stands much up over there, just behind the US there. What is remarkable to note is that from the whole of African continent, it's only South Africa. Nobody else over here in this list. And you can see that the virus is concentrated in certain parts of the world, as you can see in North America, Europe, and of course, India. But then this is from October 9. Today at 10 a.m., we have more than 1 million deaths, as you can see. But again, the deaths are concentrated in certain parts of the world. Europe, as you can see, Central and South America. In the US, of course, it's mostly in the East Coast. Look at in Africa, very few. Look over here in India, Indian subcontinent, the death rate is very low. As I said, whereas in the world as a whole, the fatality rate is 3, in India is about 1.4. Why is it so? So I think there are a few issues that one can talk about. Is it due to the microbial load? these two continents, and I call Indian subcontinent also a continent there, these two continents, as you can see, have been subjected to a lot of microbial load. And we must have therefore generated a whole variety of memory cells. Our best sort of, the bet was then when we saw in Dharavi of Mumbai, everybody thought that Dharavi is going to be really infected very large. And there is a 800,000 population there living in a very small area there. But the infection rate in Dharavi was very low. So was the death rate very low. So I think the two things that come to mind is it microbial load, which is responsible, and is it due to the hygiene hypothesis? ऐसा मैं एक लेक्चर में हूँ, मैं थोड़ी देर में करता हूँ आपको फोन, मैं आप ही करता हूँ. So I think these are very clear in terms of the death rate. Let me show you this picture, which is really very nice, and I'm very fond of showing this. This is COVID-19, the whole scenario as you see. The SARS-CoV-2 starts its journey in the nose, in the mouth, or in the eyes, and travels down to the alveoli in the lungs. And you can see here the alveoli lined by the capillaries, and the capillaries are the ones, the site where there's a gaseous exchanges take place. The carbon dioxide brought by this blood over here, it's called exchange, and they take the oxygen there. The alveoli are lined by two types of cells, the type 1 cells and type 2 cells. The type 2 cells are very important because type 2 cells are the ones which excrete surfactant. It is a substance that lines the alveolus and prevents it from collapsing. This is what happens in the healthy state. So you really do have the gaseous exchanges over there. Now, what happens if the virus has entered? Now, here is an individual, we can call it as asymptomatic or a very mild kind of infection. You can see the spike over here, the spike proteins of the virus covering the coronavirus bind the AC2 receptors primarily on the type 2 alveolar cells, allowing the virus to inject its RNA. The RNA actually hijacks the cell. It is like a dacoit coming to your house, makes you, keeps you as a hostage, and then asks you to get some more of them inside the house. So the same way over here, the RNA hijacks the cell, telling it to assemble many more copies of the virus and release them into the alveolus there. This is what happens in the asymptomatic or a milder state. What happens in the moderate state? In the moderate state, after infection, the type 2 cells will release inflammatory signals that recruit macrophage kind of cells, the immune cells. Macrophages will release cytokines that will cause the vasodilation, which allows more immune cells to come to the site of injury and exit the capillary there. And fluid starts to accumulate. This fluid starting accumulate is a dangerous stuff because it now dilutes the surfactants that are released by the type 2 cells, and therefore the alveoli start to collapse. So therefore the gaseous exchanges cannot take place, and the person starts to feel breathless over there. Also, it happens that at this stage, the neutrophils are recruited to the site of the infection, which is much more when you go to the severe stage over here. The neutrophils start to appear at the site of infection, and they release the reactive oxygen species to destroy the affected cells. So you can see that eventually type 1 and type 2 alveolar cells are destroyed, leading to the collapse of the alveolus and causing what we call ARDS, the acute respiratory distress syndrome. If the inflammation becomes severe, as you can see over here, the whole alveolus is full of the fluid, so much so that it can now enter the bloodstream and travel elsewhere in the body and causing what is called SIRS, the systemic inflammatory response syndrome, which can lead to septic shock, multi-organ failure, and that's the stage when the individual would actually be very difficult to save. So this is the whole scenario, the spectrum of COVID-19 infection as we see here. Now, AC2 expression we have all talked about. This is the picture trying to show you the AC2 expression in organs and systems most frequently implicated in COVID-19 complications there. Then you can see that they're much more expressed on intestines, kidneys, and the testes there. And they're also expressed on the lungs, of course, but not as much as you can see in the intestines. but the expression is much higher in the alveolar epithelial cell that we talked about. And that is why the alveolar cells actually get destroyed there. But you can see that they get expressed also in the brain, in the heart, the liver, and the intestine. And you heard about AC2 increased in patients with heart failure, and a lot of people, as we are seeing, dying due to problems in the cardiac issue there. So these are actually the key phases of disease aggression, that starting from the upper respiratory tract infection, the pre-symptomatic, asymptomatic stage where the SARS-CoV-2 has just tried to enter into the epithelial cells through the AC2. The second stage comes the symptomatic, the early phase, virus infects the AC2 expressing type 2 alveolar epithelial cells, and the patient exhibits symptoms of pneumonitis. And then this severe stage, which involves disruption of the epithelial endothelial barrier, complement deposition, and hyperinflammation, the later phase. So these are actually the three phases that we're talking about, which is very clearly shown over here. If you look at the prognosis of the COVID-19 infection, and you see this contagion, the stage at which you actually get infected, and you won't even know that you get infected until the symptoms appear. The symptoms appear something like day 5, day 4, or day 5. And in each of these 4 categories that we are talking about, no symptoms, mild or moderate type, the severe type, and the ones that really are in the more severe stage over there, no symptoms in the first 4 days. You won't even know that you got infected, although you have been infected. This is the day when you get the first symptoms. Patients who have no symptoms, they become immune, they raise neutralizing antibodies, and therefore they remain healthy. Many of them don't even know. And you've seen that the surveys that have been done, antibody surveys that have been done, a lot of people did not have any symptoms there. The second category, they have very mild ones, but they also eventually become immune. No deaths in these 2. In the severe ones, they come to the hospital, they had to be hospitalized. So we call these 2 groups as a hospitalized group. But this one probably needs only the oxygen support and they become immune. Many of them actually survive. The death rate in them is rather low. It is this variety over here which needs to be put in ICU. We need to be put on the ventilation over there, and about half of them actually die. So look at the COVID-19. There's an unprecedented growth in scientific inquiry on this. And some of our friends looked at the PubMed. Until 2nd September, COVID-19 had 47,000 papers in previous 8 months, which is about 30 times more than the human genome in 20 years at that time. So we're talking about 6,000 papers per month. So, you have to be sitting on the edge of your chair to keep yourself updated with the current knowledge as is going on. And more than half of these papers are in relation to COVID immunity and vaccines there. This brings me to the question of the immunity. And I want to take you to this immune response stadium and talk to you only about 3 players and not the 11 players that we see in the IPL. Our player number 1 is the MSC. It is a batsman which must see a ball, is your pathogen, sort of divided, sort of broken down to epitopes, the MSC peptide complex on the APC seen by the CD4 T cell of the host. Because CD4 T cell is a headquarters cell. It will then present or sort of stimulate either the helper, give help to the cytotoxic T lymphocytes or the B cell. This is in simpler terms what we have the immune response stadium. And then there are 2 components of the immunity: the innate immunity that you already know, the pre-existing ready to attack, and the adaptive immunity, which is stimulated by exposure to microbes and which is more potent there. But what I wanted to say in this particular slide is, because you are exposed to millions of pathogens in a country like India or African continent, the microbial load is very high. The MSC is also very highly polymorphic. Our studies in AIMS have shown, and so have the studies shown in Africa, countries where the microbial load is very high, the polymorphism of the MSC is also very high, because the basic biological function of MSC to present the peptide to the immune cells. And this is a clear picture you have already seen in Abul Abbas' book. I've copied from there. Innate immunity and adaptive immunity, the cell types which take part, the epithelial barriers. Here it only is reactive to microbes, whereas the adaptive immunity, as you can see, is reactive to microbes, non-microbial components, as well as to self as well. But the adaptive is the one which is stimulated by exposure to microbe, and it is more potent there. And this is what you see in COVID-19 when you're looking at the test. The test that you get done is the PCR-based test over there. This is the time when you have the symptom, about a week or 5 days prior to that, you got the infection, but the peak of the virus comes on the day of the infection, and then this peak remains, and you're looking at the nasopharyngeal swab or the bronchoalveolar lavage or the virus from the respiratory tract. So I think the best time to get your test done would be in the week 1 of the appearance of the symptoms over there. And then you also know the IgM and the IgG and antibody. This is the spectrum. We will talk about it more as we go through our other slides. But I made a star over here. The star in terms of the SARS and the MERS. You see, the viral load of SARS and MERS actually peaks at around day 7 to 10 after the onset of symptoms. Here, the virus load appears on the day of your symptoms over there. So here we had a window period there after the symptoms. You take about 7 to 10 days when the virus goes up. Here it's right on the same day. This is the difference between the two. So if you look at some facts of COVID-19 immunity, there is an elevated inflammatory response, but response to anti-inflammatory compounds is mixed. There's a lymphocytopenia. Unlike other infections, the lymphocyte number is down in COVID-19, similar to what we see in HIV infection and AIDS or in hepatitis. Patients who are able to recover this number by days 7 to 10 develop only a mild or a moderate form of the infection, and they eventually recover. But those who with consistent lymphopenia develop more severe disease and often go on to develop a very critical phase. T cell activation, and there is T cell exhaustion, there's a hyperfunction, and we'll talk about that. We have also learned about the cytokine storm, which is an expression of an exaggerated immune response over there. Plasma blasts, the most, the paradigm is that you see plasma blasts in the peripheral blood, you see antibodies in the hospitalized individuals, but they are still sick. And we want to know why. Does the convalescent plasma has any role? But what is more important to an immunologist is there a typical immune response in COVID-19? And the answer is yes and no. This is a very important paper which came up from the group in Sloan Kettering. These are the people who have been working primarily in cancer. And they drew a parallel between the situation that they see in cancer or in tumor growth and in COVID-19 immunity. And very important, they said there's an imbalanced host immunity to SARS-CoV-2, which drives the COVID-19 infection. The COVID-19 infection is a case of innate immunity hyperactivation and a case for adaptive immunity dysregulation. What you see in the innate immunity here is a cause of severe COVID-19 infection. These patients harbor an expanded population of circulating monocytes that secrete those cytokines, the two very important cytokines we're talking about, IL-6 and the IL-1 beta there. So an innate immune-mediated cytokine storm is the one that you see. And there's also a case of adaptive immune dysregulation where the viral dissemination and circulating viral RNA in the peripheral blood is strongly linked to disease severity. Hence, an intact adaptive immune response is the key to its clearance. And this will become clearer. But how do we achieve this? How do we make sure that this dysregulation moves to a normal state, and we sort of make sure that this is not in the hyperregulation? There are a few possibilities that are emerging now, and probably towards the end of the talk, I will touch upon that. This is what happens in the innate immune regulation of antiviral defense and tissue toxicity. In the normal immune homeostasis, you know that the innate immunity works with the PAMs and the DAMs over there, picked up by the macrophages, and then you have those cytokines, the IL-6 and IL-1 beta there. And this is what happens in the normal immune homeostasis. What is happening in acute lung injury is that the viral-derived DAMs and PAMs activate tissue resident macrophages. They activate the innate immune pathways through either the TLRs or the NLRP3, and you already know of those pathways, inflammasome activation and others. Production of two very important cytokines, and maybe others, but the two very important are the here in COVID-19 are the IL-6 and the IL-1 beta, leads to the recruitment of neutrophils now. So you see that the neutrophil number going up, and the CD8 T cells which control the viral growth. But it also induces tissue damage, leading to alveolar flooding, and that you already saw over there. People have been trying to use this monoclonal antibodies to try and see if you can block this IL-6. Let's talk a little bit about the cytokine storm. The cytokine storm that you see in viral infection of COVID-19 and the cytokine releases that you see due to immunotherapy in cancer by CAR T cells. Here is what you see in terms of CAR T cells. During CAR T cell driven cytokine release syndrome, blockade of this two cytokines, the macrophage-derived IL-1 or IL-6, limits the tissue toxicity without interfering with the anti-tumor immunity. This is not the case in COVID-19. Again, it's the same phenomenon that, as I talked about, that during viral infections, blockade of macrophage function may actually impair both innate and adaptive viral control. So therefore, you have read some studies that people who have used this anti-IL-6 or the anti-IL-1 alpha have not been very, very successful, unlike in the tumor state. So that's the difference they were talking about. Now look at this immune dysregulation that we notice during the acute and the chronic viral infections. What happens in the acute stage? During acute viral infection, early innate and adaptive immune function leads to viral suppression. You have this innate immunity, and that they eventually will go down and taken over by the adaptive immunity, and there's a long-term protection conferred by that because you have the memory T and B cells formed, you have the antibodies over there. So the late adaptive immunity due to cytotoxic T cell, helper T cells, and B cells and antibodies. This is a sustained adaptive immunity that you normally see in the acute stage. But in the chronic stage, this is what we are seeing in COVID-19. During chronic viral infection, persistent viral leads to T cell depletion. There's a T cell exhaustion, while triggering the ongoing innate immune inflammation. Innate immunity doesn't go down. It remains in a state of activation there, the hyperactivation. Therefore, the virus is not taken care of, and you see there's a viral dissemination is the key driver of severe disease there. This is exactly the difference between acute and the chronic state. Now, how do we balance innate and adaptive immunity to combat COVID-19 infection? After all, there should be something that we could do. So this paper talks about three very important pathways to improve anti-COVID-19 immunity. It may be necessary to enhance the antiviral sensing through activation of interferons, and I'll talk to you about the interferon towards the end. That you could, again, the role of type 1 interferons over there, which could sense the PAMPs and the DAMPs and therefore activate the immunity. Or you could do the enhancement of the adaptive immunity through these pathways, and I don't want to go in details of that. You could actually look into this study. They looked into IL-7 and NAC, and all of those things, enhancing adaptive immunity, by rescuing T cells from exhaustion or exhaustion-dependent cell death. Or the third, as we have already talked about, blocking macrophage-dependent cytokines by the monoclonal antibodies there. So these are the three mechanisms that they were talking about. This paper came up in July. You know, in COVID-19, we are really changing our stand every month, I would say. Now, if you look at the immunopathology of COVID-19 that I've talked to you so far, immunopathology tells you that there's a lymphopenia. There is a lymphopenia of all of these cells, the CD4, CD8, the B cells, the NK cells. There's a T cell activation, and there is a lot of the cytokines. You can look at this T cell activation by these three, or all of these five markers on the cell surface of CD69, CD38, 44, and others, and they release this cytokines, and therefore T cells remain in a state of activation. There's a T cell dysfunction as well because of the TIM3 going up, the NKG2A going up over there. This leads to T cell exhaustion and even NK cell exhaustion there. On the other hand, you also see abnormalities of granulocytes and the monocytes there. The neutrophil number going up, while the others, all of the other cells are going down. And there is that we saw increased production of cytokines, several of them, but we are anyway talking more about IL-1 beta, IL-6, and the IL-10, but others also go up over there. And what you also see is the total antibody. The whole of immunology is right there in this immunopathology of COVID-19. This is a very important paper I just saw 2 days ago. This appeared on October 7, although I think the data, this paper was submitted to Cell in June, revised in August, and accepted by them in September 30. It came up in October 7. This is a very nice study, and I really enjoyed going through this. This talks about the imbalance of regulatory and cytotoxic SARS-CoV-2 reactive CD4 T cells in COVID-19. They have the study on 18 non-hospitalized individuals and 22 hospitalized, of which 9 were in the ICU, and they used the single cell RNA-seq to look at the SARS-CoV-2 reactive CD4 T cells and transcriptome analysis of more than 100,000 single cells over there. And what they're finding is summarized here, the messages of the study, that early in illness, hospitalized COVID-19 patients develop a strong cytotoxic T follicular helper cell response that can potentially kill the B cells and therefore reduce the antibodies there. In fact, they've shown this picture of cytotoxic T follicular helper cells going up and the antibody titers going down. They also found remarkable heterogeneity in the nature of CD4 T cell subsets reactive to SARS-CoV-2 with differing severity of COVID-19. But what I wanted to really talk about was the group of patients mounted impaired Treg response. As you can see, the Tregs going down in the presence of a very strong CD4 CTL response there. I think a study like this will have to be done in a longitudinal stage. So you not only just take at one time point, but if you take at different time points and then see the way the immune system is going. Again, this slide actually summarizes the potential mechanisms of SARS-CoV-2 induced immunopath, as we talked about, that there's a depletion and exhaustion of the lymphocytes, and they've talked about 4 different phenomena because of the AC2 going into the T cell, they entering the T cell, impaired the T cell depletion and exhaustion, lymphopenia due to the damage to the spleen and the lymph nodes over there, and the lactic acid going up over there leads to T cell impaired and dysfunction. And if the T cell numbers are going down, the lymphopenia is going around over there, the microbial infections will go up, and this will lead to activation recruitment of neutrophils there. So people are actually looking at the neutrophils, the lymphocyte ratio, and the cytokine storm we have talked about. And then those of you who are very interested, it's a very nice paper which came up from China, but a lot of support from UPenn. And the antibody-dependent enhancement, which actually makes sure that the virus actually enters the cells and the disease can progress with the poor outcome. The clinical implications of SARS-CoV-2 induced immunopathology that we talked about, that if the lymphopenia sustains, microbial infection goes up, the effect of lymphopenia on microbiota infection, effect of the elevated cytokines would lead to multiple organ dysfunction. This is what one sees in the late stage, in a very severe stage, that there is a multi-organ function there. I want to talk about a little bit on the, I know I started writing for Hindustan Times, and I thought this is the time to really discuss some of our own studies. And I wrote this first article, came up in April 1. Can India be an outlier in the spread of infection? Because at that time, Our numbers in hospital were very low. Our numbers on the ventilator were very low. By the way, it is still the same. Although the Indian authorities created a whole lot of these hospitalized beds and all that, they remain empty. And the ventilators are also empty. So my hypothesis at that time was the broad-based immunity in the Indians due to the high microbial load, as we have talked about. And you saw on that John Hopkins dashboard. Our friends in the other field, they talk about food habits and the environmental factors. The kind of food that we take would also probably be immune activating. But I talked to you about the extensive diversity of HLA in the population of India with novel genes and unique haplotypes. This is a study that we've done for over the years, and we found that the diversity of HLA in the population of India is extremely high. This is a very popular slide that I normally show, the diversity of HLA-A2. There are about 300 molecular subtypes known, and look at the Caucasians, only one subtype, O201. And as you go to the other populations, there may be 2 or 3 subtypes, but the population of India, it is a heterogeneity being very high. I call it as a khichdi, and I call this one, the 211, is actually a novel allele that is found in the population of India, not found in other ethnic groups, certainly not found in the Caucasians. And there are many others. Of course, we found it in the Hungarian gypsies. And this slide, of course, summarizes some of those studies that we've done, and people can really look into those. In almost every allelic family, we found those remarkable diversity in the population of India, and not as much in the other ethnic groups. So this is definitely due to the microbial load that we have in the population of India. And there are a few studies being done already on HLA and SARS-CoV-2. COVID-1 was done earlier, had shown some of those data there, but now people are beginning to come up with this data. By the way, by now, International HLA Genetic Consortium has also been made, and they will, I'm sure, very soon come up with data, because the feeling is there could be some people with a particular allele or a haplotype more susceptible. This is another very important paper talking about the immunotypes and association with clinical severity. Again, a UPenn study taking 3 groups of people, healthy donors, recovered donors, and the COVID-19 patients of various grades of severity over there. And they looked at the high dimensional clinical data, high dimensional flow done, and talked about 3 immunotypes. And really, because of lack of time, I will not go into details of this actually. And they have used this terminology UMAP. UMAP is a uniform manifold approximation and projection approach that they have used. But what is more important is that there are 3 immunotypes are defined that are linked to various presentation. And you can see that the severe disease is mostly in the immunotype 1. If you're born with it, if you have this immunotype 1, and no severe disease over here. And here, of course, there's a negative correlation between the immunity and the severe disease. So there is it seems to be apparent good correlation between features, as you see in the hospital, and treatment outcomes with the presence of specific immune cells. Again, a very important study which came up in September 4. What happens in the asymptomatic individuals? People are talking about asymptomatics. What is the clinical and immunological assessment? I have seen only one study, which is really very nicely done on 37 asymptomatic individuals in China without any relevant symptoms in the preceding 2 weeks and during hospitalization. This group had a longer duration of viral shedding than the symptomatic group. And the virus-specific IgG levels in the asymptomatic group are significantly lower. This is a very important point. And of course, this data suggests that asymptomatics have weaker immune response to SARS-CoV-2 infection. Whether they are infective or not, I think that's a debatable point. So if you look at the path to adaptive immunity in the data summarized until September 11, this is a paper in JAMA, and I would want all of you to read about that it talks about not only the B-cell antibody response, but also the T-cell antibody response, and the timing of the appearance of these antibodies over there. They're both IgG and the IgM. We have been only talking of IgM and IgG, but IgA also could have a very important role, and the timing when the IgG comes. And look at this CD4 and the CD8 cells, and the memory cells which sustain, and the memory cells and the B cells which also sustain and, and remain. So that if there's an anamnestic response to reinfection or the vaccination there, this is a long-term adaptive response. This paper really summarizes the take-home messages from this are really very bright. And you could actually look into this paper more. I want to go on to the next, in the next 5 minutes, only talk to you about two things. One is on-spot detection. People are talking about, should we open up travel? Should people be going international travel or not? If they want to go on travel, is there a test that we have for on-spot detection? And you read from time to time a lot of chips being made and all that. And the second thing that I will talk to you about, the alternate therapy. Does immunotherapy have a role? And I got this particular slide of a dog being trained. You see this a Springer Spaniel being trained at the University of Adelaide, Australia, to detect signs of coronavirus infection in human sweat. And they feel, and the data that they have so far is very strong, that you could use these dogs. If that could be done, that will be really, that could open up international travel as well, because you could then identify people upon arrival who may have this. But this is all under trial stage. But I want to talk to you about the therapy. Here is this generation of glucocorticoid resistant SARS-CoV-2 CTLs. Everybody is talking about antibodies. Everybody is talking about neutralizing antibodies. But do CTLs have a role for adoptive T-cell therapy of critical COVID-19? This is a study where they expanded CTLs against the structural proteins of SARS-CoV-2, particularly the RBD of the spike, by using a cocktail of cytokines. That they've used these two types of cytokines, and they've taken the recovered donors and the healthy donors over there, and they've found that the expansion was possible only for the COVID-19 recovered individuals and not for the healthy, rightly so. And they could have the 1,000-fold more expansion of these CTLs, actually, both with the IL-247, or if you use the 27 and 15 there. So since corticosteroids are increasingly being used to treat the critically ill COVID-19 individuals, the corticosteroid receptor gene in this study was very efficiently, the NR3C1 had to be inactivated. They've done that using the CRISPR-Cas9 gene editing, the guys who got the Nobel Prize. So this is a beautiful study. You could actually opens up a channel that you could use the cytotoxic T lymphocytes for adoptive therapy. And the last point I want to talk about the interferons and the COVID-19. And you saw in my first slide over there somewhere, I've talked about the interferons, which are a group of, as you know, signaling proteins made and released by host cells in response to several viruses. And we immunologists normally are very fond of talking about gamma interferon. But there are two major classes of interferons, the type 1 and the type 2. Here I'll be focusing more on the type 1 interferon, particularly the IFN alpha, which is known to influence innate and the adaptive immune response to the viruses. Interferon alpha is primarily produced by the plasmacytoid dendritic cells, whereas beta is by several subtypes. Their production is induced after the sensing of microbial products by PRRs and by cytokines. And type 1 interferons are protective in acute viral infections, but have either a protective or a deleterious role in bacterial infections. So here we are only talking about the acute viral infections. What it turns out is that there could be neutralizing IgG autoantibodies formed against type 1 interferons, and that have actually been seen in diseases like lupus, and diseases what we call APS1, the autoimmune polyendocrinopathy syndrome type 1. So this particular studies are trying to look into the role of SARS-CoV-2 virus infection and in influencing the development of COVID-19. And this, of course, is a cartoon trying to tell you how type 1 interferons control the innate and the adaptive immunity as well as the intracellular antimicrobial systems through the plasmacytoid dendritic cells, which have seen the PEMs and the PRFs. They release this alpha interferon, the type 1 interferons, and these are the kind of things that they do. But the type 1 interferons, again, are also produced by those innate immunity cells, the dendritic cells, and the macrophages. They can also, as you can see, influence the adaptive immune system as well, the T cells, the effector T cells, and the antibodies there. And this is a very important study, again, by Paul Bustard in Rockefeller, which came up in Science in September 24, where they looked at the life-threatening COVID-19 autoantibodies, again, type 1 interferons, and patients with life-threatening COVID-19. They actually found that there are autoantibodies formed, mostly in males, and also more in individuals more than 65 years of age. And the take-home message from this study is more than 10% of patients hospitalized who have a critical COVID-19 had neutralizing IgG autoantibodies against the type 1 interferons. This is compared to 0 in those with mild or the asymptomatic disease. And mind it, the number is not small. 663 here, 987 here, and just about only 0.3 in healthy individuals. And there was preponderance of males with severe COVID-19, and we don't know why. The paper actually talks about it could have a genetic component there, because 2x in women, with 2x, there could be an X-linked recessive trait, because they actually saw a female patient with a rare condition that silences one X chromosome, and she developed a severe COVID-19, and she had autoantibodies there. So the question is, could synthetic IFNs given before and after an infection with COVID-19 help block the virus? And I think this is something that the whole field is moving towards, looking at alternate medical tools. And this is again an article that I wrote for Hindustan Times, came up in October 17, where I discussed about alternative medical tools of COVID-19. And these are those three: the designer antibodies. Our friend Donald Trump was given this, the company Regeneron, and the confidence level of this company and the doctors treating Trump was so very high that they gave him 8 grams of this. And because the Regeneron feels that it can reduce viral load, improve, and it is improving the non-hospitalized COVID-19 individuals, and actually benefiting those who do not make any antibodies. Again, the other alternate tool is understanding mechanisms driving high infectivity of the virus. And I talked about the linoleic acid as a tool in a tailor-made pocket in the RBD of the virus. And this is what the cryo-EM data shows. This is a paper which again came up in Science in September 21. And I already talked to you about the type 1 interferons. This could become another tool. So until the vaccines arrive, there are other tools, and the immunologists are right on their guard to look into these issues that one can do, because it could be the alternate way. This is my last slide, but I want to leave you with several unanswered questions, and they're all shown over there. I don't have to go through each one of them. I will leave this slide. Whether patients who recover could develop long-lasting immunity against reinfection, we don't know as yet. Whether the reinfected people will develop less severe disease compared to those who were previously non-infected. And of course, we have seen a few reports so far of the reinfection, and the infection was not very heavy, but you don't know. And then the whole lot of those questions in the asymptomatic individuals. Thank you very much for your attendance. And I will be very happy now to take up any questions that you might have.
Thank you very much, Professor Mehra, for a very elusive talk on immunity and COVID-19. We have few questions in the question answer box, and some of them have been seen by Dr. Shiv Pillai as well. So what I think is the next, I don't know whether these people, they want to ask this question these questions to you or not?
The
Because
Shiv wants me to share my email. Kindly send my email to him immediately, and I will, of course, write to him. Yeah.
all right, I will do that. Uh, mm, what we can do is uh, I can still ask you a few questions which are there in the open box. Uh, okay.
I'm sorry if I answered the questions and they were meant for Narendra. I just saw them come to me, I answered.
Oh, that would be really great. Shiv, I give him my talk, and your talk is following me, and you're also talking about immunity in COVID-19.
I
And
was trying to send you a question and I saw these questions coming. I couldn't put in my question. So I just answered what I saw.
But that's okay. I can still bring out those questions because they are interesting. Some of them are very interesting, and I think Shiv will also go and elaborate on them. The first question is by Srijita Roy. This is why does antibody titer of IgG and IgM decrease after a few weeks of COVID-19 infected persons?
this is a very important question that everybody is asking. Of course, we do know that by 3 months or 4 months, the antibody levels go down. And this is something that I really discussed in my last article in the Hindustan Times. There's nothing to worry about. Antibodies are known to go down after every viral infection. But there are memory cells, and we need to really find out tools of looking at these memory cells. And we have actually been struggling even in the organ transplant field, looking at where we want to look into the memory B cells in the peripheral blood of people who have been given an organ transplant, because then we can know these are people who are more likely to develop antibodies there. But any immunologist eminence, and there's a very eminent immunologist who is going to talk just after me, he will talk to you that there will be a small group of long-lived memory cells that remain over there. And therefore, that slide that I showed you, that the anamnestic response or the secondary immune response, which could come due to the reinfection or due to vaccination there. So the vaccination will become even more important because you do have those long-lived cells. So antibody is going down, but then the CD4 T cells as well. Nobody is talking so far about the cytotoxic T cells. We do have a sort of small window or small numbers of memory cells, both in the T compartment as well as in the B, and they remain over there. So antibodies do go down, nothing to worry about.
So there is one more question which has not been answered yet. It is, what are your views on herd immunity?
I had in that slide, and because of lack of time, I was very scared of the chairman of this session. He might say that you're going over time. Herd immunity is something that everybody is talking about. Herd immunity can only come through vaccines, or it can come, there's a group of people who say, oh, let the infection grow. So that means that you want at least half of your population, 130 crore Indians, and you want half of them to get infected, and then you can probably get herd immunity. So herd immunity will only come if we have a very effective vaccine. What I'm saying is that there are now hundreds of vaccine under trial. There are at least, I think the last I saw about 15 or 16 that are now in the phase 3 trial over there. We might get a vaccine, but please be at guard. We do not know the effectivity of the vaccine that we may get. WHO really keeps the vaccine effectivity to be nearly about 70. Although sometimes they talk about even if 50 is enough. I think and you as immunologists would know that the effectivity of a vaccine should be 70% and above. That can create a herd immunity for us. Herd immunity is not going to come like this because the price that you will pay by infecting half of your population is very, very heavy. A lot of people will die.
All right. There is another question very interesting to me. Actually, it says, how was the 2 meter distance in the social distancing was arrived? Whether there is any scientific basis on this or what?
Oh yes, there is a scientific basis of those three things that we're talking about, which we are talking in the form of the social vaccine, the the mask, the hand hygiene, and the social distancing of 6 feet or 2 yards, and our PM says do gaj ki duri. Now, because it's a droplet infection, and there are studies done with very high-powered cameras in Japan, which have shown that even while talking, you may be actually those very fine particles, and they remain in the air, and and they can really travel up to 6 feet. And I think this 6 feet, it came up. The terminology came up in March, April, but I think it has sustained, and everybody says the 6 feet is the one, the minimal distance there. So I think there is a scientific basis of that. People have used the high density cameras and looked into those droplets and having those viruses there.
Yeah, the droplets, they cannot travel too far, so that's why they drop on the in between. So that's why this was a scientific basis. A lot of physics has gone into that to show that they are heavy, they drop, and they cannot cover 2 meter distance. So that's why that was, anyways. So there is another question: when antibody titer goes down, then how does plasma therapy works? I mean, this
This is another very important question. And I just reviewed a paper. Somebody was trying to do meta-analysis or a review article on plasma CP, I mean, convalescent plasma there. Contrary to our belief, convalescent plasma has not been very successful. And partly because a lot of these people, as you saw, that their antibody titers may be low, unless you do that analysis and find high titer antibodies. This is what Regeneron has done. Regeneron made those humanized antibody in the mouse and the other antibody, because it's a cocktail of antibodies that they make. And the other antibody they took from a recovered individual were high titer IgG antibodies. So unless you have those people screened, it's not that just anybody and everybody is not recovered, because a lot of people who may have very low titer antibodies and the convalescent plasma in them may not have. You saw that Donald Trump has been given everything. I mean, Regeneron, and the company actually started saying that the Regeneron, the Regen.
Sir, your voice is cracking.
What
Okay.
is that?
What I read was there's a very interesting study about convalescent plasma. It said that if you give convalescent plasma within first few days, like 4 or 5 days, then it shows effectivity. Whereas if it crosses 7 days of infection, then the efficacy goes down.
Absolutely
So,
right. Absolutely right. Because CP was earlier, when the trial began, it was being given to the hospitalized people, particularly the ones who were on the ventilator there, and there was no response. Rather, it was doing bad. It was doing harm to them more. So if it is given to the early stage, now the early stage is what? Asymptomatic stage or the mild state? Should you give? Because the guy is already, and the guy is asymptomatic, he won't even know. And the asymptomatic and the one with the milder one, we say, okay, you go to quarantine and we will watch and see. So, the CP has lost its charm as it began, since the time it began.
I think it's
so
going to be
I will
replaced.
ask.
Yeah, sorry. I think it's going to be
Yeah,
replaced.
just one more question. And
Maybe, sure.
there are many questions. We'll ask later on with once Shiv has given his talk, because it's almost same. But I'll ask one question more to Dr. Mehra. This is microbial load hygiene hypothesis. Why are municipal workers who are cleaning roads and collecting garbage not falling sick?
This is what? I mean, you are actually supporting my hypothesis. The microbial load, actually, when the infection started to grow in Mumbai, I was very worried that Dharavi is gone. Dharavi, 8 lakh people living in an area of 1.5 kilometer there. Look at the numbers, very few. Look at the deaths, very few. So and we have seen it now that the pockets where the infection rate is very high, the low socioeconomic status and all, the hygiene is very low, the infection rate is low. So it has an immunological basis over there. Some of you have read about the hygiene hypothesis, which was started actually by Saint-François Bach, Saint-François Bach in France, primarily in looking at the preponderance of diabetes around the world. And he saw that in Scandinavia, for example, in Finland, the incidence of diabetes was so very high, infections are very low. And you go to Africa, the autoimmunity is very low. So it's just the reverse of that. Infections are very high and all. So hygiene hypothesis and microbial load are two factors which are protecting India in terms of the death rate. Of course, our infections are growing because as we opening up, our infections will grow. But most of them will remain in the asymptomatic stage. Most of them will remain in the milder state over there.
Thank you, sir. Thank you very much for this very nice talk. And I move on to the next talk. Thank you very much.
There, and after Shiv's talk, and if there is any more,
yes.
I would join Shiv. I will learn from Shiv any more to be answered.
Very kind of Shiv. We look forward to that discussion because it's going to generate a lot of discussion on COVID after Shiv's talk. So please stay on, and we'll like to have you again on board. Thank you very much. So I need to invite